All right, so let's continue talking about heredity. So earlier we talked about dominant versus recessive alleles. I'm going to introduce some more vocabulary for you. If you have both the dominant alleles, so say that you have brown eyes and both parents gave you the allele for brown eyes, you are referred to as homozygous dominant. Homo means the same, and dominant, of course, means dominant. So if you have two dominant alleles, you have the same two dominant alleles, you are homozygous dominant. If you have one dominant allele and one recessive, you are heterozygous because the two letters are different, but you are still dominant, so you're still referred to as dominant. And then if you have both recessive, you would be homo, again, because it's the same, recessive, because you have two lowercase letters. Oftentimes when you talk about like breeding cats or dogs, you'll hear terms like purebred or hybrid. So if you are purebred, like this, if you're a purebred German Shepherd or whatever, that means that dog is homozygous dominant for being a German Shepherd, I guess. But if you are a mutt or a hybrid dog, whatever, then you would be heterozygous because you have a mix of genes, dominant and recessive. Now, it's note that it is impossible to be homo or heterozygous recessive because you need two copies of something to be recessive. So there's no such thing as homozygous, heterozygous recessive. All right, so let's talk about how to determine phenotypes. This is the part that everyone tends to like. So we determine phenotypes or physical characteristics by using a Punnett square. So a Punnett square looks like this. It's just a square with 4 boxes in it. And on one side will be the parents' alleles. So let's say mom's on the top here, dad is on the side. And if we know the parents' alleles, we can plug them into the Punnett square and we can figure out the probability of their children having certain traits. Now remember, dominant alleles are represented by capital letters, recessive alleles are represented by lowercase letters. So let's say, for example, that mom is homozygous dominant for hair color or whatever we're talking about. So if she's homozygous dominant, I'm going to give her 2 capital letters. And then let's say that dad is homozygous recessive for something. So he gets 2 lowercase letters. Now the way to solve a Punnett square is to drag the letters down into their corresponding boxes. So this first letter A here is going to go into this box and this box. So we write a capital A in both. And then this capital A goes down into these 2 boxes as well. And then going over here to dad, this A goes into these 2 boxes. So lowercase a, and then this A goes into these 2 boxes. So lowercase a, lowercase a. Now, according to our Punnett square, this child now has a 100% chance of being heterozygous dominant for whatever trait we're talking about, because all the outcomes are uppercase A, lowercase a. All right, I'll do another one for practice sake. So let's say that mom in this case is heterozygous dominant. So uppercase A, lowercase a, and let's say that dad is heterozygous dominant too. So again, this first capital A goes into these 2 boxes. This lowercase a goes into these 2 boxes. And then the uppercase A goes into those 2 boxes. And this lowercase a goes into these 2 boxes. Now we can see in this situation that the probabilities are a little bit more different. So we have about a 25% chance of being homozygous dominant. We have a 50% chance of being heterozygous dominant, and we have a 25% chance of being homozygous recessive. Right? So if these 2 people had a child, most likely the child would be heterozygous dominant, but there's a chance it could be either one of those as well. So there are different types of Punnett squares, and for the most part, we will focus on monohybrid crosses, which is where we only consider probabilities for one trait at a time, meaning there's only one set of letters on the top and the bottom here, and we will figure out the possible genotypes and therefore phenotypes of the offspring below. Now, there are also complicated Punnett squares. These are called dihybrid crosses, where you figure out the traits of 2 things at a time. So in this example, color and shape. You do not need to know how to do this, but I just wanted to show you one. to show you how complicated the genetics can be, because like I've said before, a lot of traits that you have are controlled by more than one gene, meaning more than one pair of alleles. So this would be one of those cases, for example. So hope that makes sense.